All right, welcome. Today we're going to dive into the incredible microscopic world of enzymes. Now, you've probably heard of them, but they are truly the unsung heroes working inside every single one of your cells, basically making life itself possible. So let's kick things off with a pretty big puzzle. Think about this. Your body is like this massive chemical factory, right? It's running millions, literally millions of reactions every single second. So how in the world does it do all that without, well, cooking itself from the inside out? That's the big question we're going to tackle today. Okay, so to get to the bottom of that, we first have to talk about something called life's speed problem. And it really is a problem. It's this fundamental challenge that every single living thing has got to figure out just to, well, stay alive. So here's the deal. For you to do, well, anything, digest your lunch, build new muscle, even just think of thought, all of that depends on chemical reactions. But here's the catch. Left to their own devices, these reactions are painfully slow. I mean, we're talking glacial pace here. Way, way too slow to keep you alive and kicking. So you might be thinking, wait a minute. I remember from chemistry class, if you want to speed up a reaction, you just turn up the heat, right? So why doesn't your body just crank up the thermostat? Well, it turns out that's a really, really bad idea for a living thing. And there are three big reasons why. First, it would take a ridiculous amount of energy. You'd be completely drained. Second, and this is a big one, high temperatures would literally start to cook and destroy your cells. Not good. And finally, heat is, well, it's messy. It speeds up everything, all the reactions at once, even the ones you absolutely don't want to happen. It's total chaos. So, if heat is off the table, what's the solution? How does life solve this? Well, this is where nature gets super elegant. Instead of using the brute force of heat, it uses these tiny, incredibly precise tools. And those tools are called catalysts. Now, a catalyst is just a substance that makes a chemical reaction go faster. But here's the really important part. It doesn't get used up in the process. It's like a little helper that can do its job over and over and over again. Talk about efficient. Okay, so catalyst is a pretty general term from chemistry. But life, of course, has its own special biological version of these catalysts. And they are called enzymes. These are the tiny helpers, the speed-up artists we've been looking for. So what exactly is an enzyme? Well, at its core, it's a big protein. It's made of these long chains of amino acids that fold up, kind of like origami, into a very specific and unique three-dimensional shape. And that shape is everything. Because it's this unique shape that means each enzyme has one job, and one job only, to catalyze one very specific reaction. All right, so that brings us to the next big question. We know what enzymes are now, but how do they actually do what they do? How do they pull off this perfect match and get the job done? Okay, to get how this works, we need to meet the key players in this little drama. First, you've got the substrate. That's just the fancy word for the starting material, the thing we want to change. Then you have the product, which is, the final result. And the most important part of the enzyme itself is called the active site. That's the special little pocket or groove where all the action takes place. And here's the beautiful part. That active site isn't just some random shape. Its shape is perfectly complementary to the substrate it's designed for. Think of it like a puzzle piece. If the substrate doesn't fit perfectly into that active site, nothing happens. The reaction just won't go. The whole thing is called the catalytic cycle, and it's basically a four-step dance. So, step 1, the substrate floats in and finds the enzyme. Step 2, it binds right into that perfectly shaped active site. Step 3, bam, the reaction happens and the substrate is transformed into the products. And finally, step 4, the products are released and the enzyme is now totally free and ready to do the whole thing all over again. It's this beautiful, efficient loop. But, science is always learning and refining its ideas. And it turns out our understanding of that perfect fit has gotten a little more sophisticated over the years. So the first idea scientists had was called the lock and key model. It's a pretty simple idea. The enzyme's active site is a rigid lock, and the substrate is the one and only key that fits. Makes sense, right? But the modern, more accurate idea is called the induced fit model, and this is where it gets really cool. In this model, the active site isn't totally rigid. It actually adjusts and changes its shape just a little bit when the substrate dines, creating an even tighter, more perfect connection. Honestly, this is the best analogy for it. Think about putting on a rubber glove. The glove isn't shaped exactly like your hand when it's just sitting there, right? But as you slide your hand in, the glove stretches and molds around it, and then it becomes a perfect fit. That's exactly what's happening with the induced fit model. The enzyme literally hugs the substrate. Okay, so let's boil this all down. What are the big takeaways here? Number one, enzymes are nature's catalysts. They're the solution to life's speed problem. Number two, they are super specific. Why? Because of that unique shape of their active site. And number three, we now know it's not a rigid lock and key, but an induced fit. That enzyme gives the substrate a little hug to make the connection perfect. And that leaves us with one last, kind of fascinating question to think about. We've talked all about how these amazing enzymes speed things up and can be used over and over. But what happens when you need to put on the brakes? How does your body tell these incredibly powerful little machines to stop working? Well, that is a story for another time. Thanks for joining me.